Welcome back to this next video in which we are discussing control of blood pressure and blood volume. Okay, so we're currently in the process of discussing the Renin-Angiotensin-Aldosterone System, and the part that we've got to now is aldosterone and what aldosterone is going to do. So we concluded the previous video by saying that when mean arterial pressure is too low, then the Renin-Angiotensin-Aldosterone system is going to be activated above its basal level, and aldosterone level within the blood is going to go up, and this is going to have the effect of raising blood volume. We also said that if mean arterial pressure is too high, the opposite problem, then you're going to get inactivation of the renin-angiotensin-aldosterone system, so the level of aldosterone is going to go below what it would be basally. Okay, and that's going to cause blood volume to actually drop down. So, how is aldosterone actually going to achieve this? Well, aldosterone is going to affect a certain portion of the kidney, and to understand how aldosterone is actually going to achieve a change in blood volume, we need to just talk about the main solute that is within extracellular fluid, not just blood plasma, but also within the interstitial fluid, which is effectively in equilibrium with the plasma fluid as far as electrolytes are concerned. So in all extracellular fluid, and I'll just remind you of what I talked about earlier, the extracellular fluid includes the blood plasma, and it also includes the tissue fluid. In all of the extracellular fluid, the main solute in that fluid is sodium chloride. Okay? Now, if we are going to increase blood volume, you can't just have all of the water. You can't just put in more water, because that would dilute the blood down. You have to have isotonic volume that you're going to add in, and therefore you need also to have sodium chloride, because that's the main solute inside there. So to increase blood volume, you have to increase extracellular fluid volume, and to do that you have to have an isotonic solution, so you need sodium chloride in there. So you can't just increase blood volume by reabsorbing more water from the kidneys. In fact, what you have to do is reabsorb more sodium chloride. The water will follow by osmosis, and then you'll have a nice isotonic solution which you can keep and retain. So the way in which we're going to raise blood volume is we're going to get the kidneys to excrete less sodium chloride into the urine, and excreting less sodium chloride, that comes down effectively to secreting less fluid, full stop. So we're going to reabsorb more sodium chloride in a certain portion of the nephron, and this is going to lead to more resorption of fluid as well, and that's therefore going to help us bring up blood volume, because we're going to distort the equilibrium. So normally, remember, of course, you'll be producing the same volume of urine as you are consuming fluid. So by reducing the volume of urine that we're producing, we distort that equilibrium, hopefully in favour of actually bringing blood volume up. And of course, lowering aldosterone level is going to have the exact opposite effect. But as always, we'll start with taking on mean arterial pressure being too low, and aldosterone is therefore increased. So which portion then of the nephron are we actually going to be affecting? Well, let me just get my picture of the nephron back up again. So, remember I said that a lot of the sodium chloride has already been resorbed by the time that we get to the early distal convoluted tubule here. Indeed, there's only 8% of the sodium chloride that was initially in the glomerular filtrate still here once we've got to the early distal convoluted tubule. And that really means that the same percentage of water will have been resorbed as we've resorbed sodium chloride. So we've reabsorbed 92% of the sodium chloride. We will have reabsorbed pretty much 92% of the water as well, because the sodium chloride is the main solute within the water. So a lot of the water will be reabsorbed as well. Now, are we going to excrete now 8% of the glomerular filtrate sodium chloride and most likely water as well into the urine? Well, no. Another 5% is usually reabsorbed in the terminal portion of the distal convoluted tubule and also in the cortical collecting duct. So in the terminal portion of the distal convoluted tubule and in the cortical collecting duct, a further 5% is going to be reabsorbed. And I mean 5% of the initial amount that's in the glomerular filtrate, not 5% of the 8%. So that means that overall, going down into the medullary collecting duct and then into the renal pelvis, usually you've got 3% of the initial sodium chloride from the glomerular filtrate, and therefore roughly 3% of the fluid. What we are now going to do, what aldosterone is going to do, is it's going to act on the cells that line the distal convoluted tubule, the terminal distal convoluted tubule here, and also the cortical collecting duct, and it's going to increase their resorption of sodium chloride and therefore also the reabsorption of water, because where salt goes, water follows is the phrase everyone uses. So that's the way aldosterone is going to work, and it means that the amount of sodium chloride we're going to secrete will drop down from 3% to much lower, and therefore the amount of fluid that will be with that sodium chloride will also drop down to much lower. So the central concept here is where salt goes, water follows. Salt is the main solute in the extracellular fluid. So if you're reabsorbing, let's say, an extra 2% here of the sodium chloride that was initially in the glomerular filtrate to take the amount that's going down into the renal pelvis down to 1%, you're also really going to reabsorb 2% of the fluid, and therefore you're going to help to increase your extracellular fluid up. So I'm just going to get another piece of paper, and then I'll actually talk about the means by which these cells of the terminal distal convoluted tubule and cortical collecting duct actually reabsorb sodium chloride and how the water will then follow. Okay, so let me draw a picture of one of these tubule cells then. So this is a renal tubule cell of either the terminal distal convoluted tubule or the cortical collecting duct. And remember, as I said at the time when I was showing you the basic anatomy of the nephron, people often judge the collecting duct as just being, cells that line that duct, and that duct's whole function is just to take the fluid that comes out of the terminal distal convoluted tubule down to the renal pelvis. But no, the cells that line that still have a resorbative and secretory function, especially in the cortical region. And the cells of the distal convoluted tubule and cortical collecting ducts, they're going to have a very similar function, especially with regards to this reabsorption of sodium chloride and the water that will follow the sodium chloride. So these are supposed to be tight junctions in between this epithelial cell and its neighbouring epithelial cells. So this gives rise to the polarization of these cells, and this is absolutely crucial concept in renal physiology. The concept that the cells can be polarized. Polarization I'm writing here. So the concept then of polarization is that there is an apical face of the cell, which we'll have as this membrane here. So this is the portion of the membrane that is actually going to face into the lumen, so you could also call it the luminal face of the cell. And there's also a basolateral face of the cell which faces the bloodstream. Okay, and the idea of polarization then is that these two portions of the cell membrane, the basolateral portion here and the apical portion here, are going to contain very different integral membrane proteins, and that integral membrane proteins in the apical portion of the membrane cannot just diffuse into the basolateral portion of the membrane because there is this obstacle, which are the tight junctions. And we're just looking at a two-dimensional picture here which doesn't really do justice to the tight junctions. These are like belts that are going to go completely around the entire epithelial cell. Okay, so if we had the epithelial cell as a three-dimensional thing, the tight junction will go all the way around it like a belt, and it fills in the gaps between this epithelial cell and its neighbour. So if I had a neighbouring epithelial cell here, the gap between this epithelial cell and its neighbour is filled in by the tight junction. So the tight junction spreads and fills in all the gaps between the epithelial cells and truly separates the apical face of the epithelial cells and then the basolateral face of the epithelial cells. And it means that proteins that are in the basolateral portion of the membrane, they can't get into the apical portion, because as they diffuse up, there's this physical barrier, okay, which they just can't get past. And that means that you can put different proteins in the basolateral membrane and different proteins in the apical membrane, and this is the utter central principle as to how these cells can overall move something from one side to the other side, from the luminal side into the basolateral extracellular fluid. So a very important concept, polarization. So we want to move sodium chloride then from the apical side to the basolateral side, and then the water will follow. So the way in which we do this is on the basolateral side we put all of our sodium potassium ATPases. Now all cells have sodium potassium ATPases which maintain the concentration gradient of sodium and potassium across the cell membrane. However, the special thing about these renal tubule cells is that they're going to have sodium potassium ATPases localized specifically into the basolateral membrane, not into the apical membrane. Again, this is going to be the basis for how sodium potassium ATPases can be useful in transporting molecules from the apical side to the basolateral side. So remember, the sodium potassium ATPase, it's a primary active transporter. It moves 3 sodium ions out of the cell and then 2 potassium ions into the cell. Every time it does that, it will consume a molecule of ATP. So that's why it's a primary active transporter. It uses the energy that it gets from the hydrolysis of ATP, a chemical reaction, to actually drive this movement of ions against their electrochemical gradients. Okay, right. So we have sodium being moved from the inside of the cell to the outside of the cell. This creates a substantial sodium gradient across the cell membrane. So sodium concentration extracellularly is usually around 145 millimolar, between 135 and 145, I should say, more properly. Intracellular sodium concentration, meanwhile, is usually closer to 12 millimolar. So there's usually around a 12-fold concentration gradient of sodium across a cell membrane. Now, in the apical extracellular fluid, again, the sodium concentration will be similar to this, because the apical extracellular fluid, the filtrate of the nephron, is going to have a similar constituency or consistency rather, and all constituency, both words work, to the extracellular fluid over here as well, because of course it is just a filtrate of extracellular fluid and all the solutes remain the same. So now, how can we get the sodium ions in the apical extracellular fluid to come into the cytoplasm and then get them over here? Well, we don't need to use an active transporter at all, because we can just put a sodium channel in the apical cell membrane, and now the sodium ions will just move from the apical extracellular fluid into the cytoplasm of the cell, because they're going down their concentration gradient, because the concentration here will be similar to over there, because they're pretty much isotonic, and also it will be going down its electrical gradient as well, because remember, the electrical potential difference across the cell membrane is generally negative, generally around negative 65 millivolts. So that means the electrical potential intracellularly is usually 65 millivolts lower than the electrical potential extracellularly. Sodium is a positively charged ion. It likes to be where the electrical potential is lower, which in this case is intracellularly. So both the electrical gradient and the concentration gradient are favouring sodium's movement inwards. So all you need to do is put a sodium channel on the apical membrane, and sodium will move into the cytoplasm of the cell from the apical extracellular fluid, which is the glomerular filtrate, or what's left of it. So the channel that we specifically put in the apical membrane of these distal convoluted tubule cells and the cortical collecting duct cells is known as the ENaC channel, which stands for the epithelial sodium channel. So of course this is an epithelium in the kidney, so the epithelial sodium channel. So it's a perfectly sensible name. Right, so again, coming back to the concept of polarization, the ENaC channels are put in the apical membrane, the sodium-potassium ATPase is in the basolateral membrane. So now look at the beauty of this. You're going to get sodium ions coming into the cytoplasm from the apical extracellular fluid, and then on the basolateral membrane they're going to be being moved by the sodium-potassium ATPase into the basolateral extracellular fluid. So look what we've overall done here. We've moved sodium ions from the apical extracellular fluid to the basolateral extracellular fluid. Now there is a problem with that. You are moving positive charge from the apical extracellular fluid to the basolateral extracellular fluid. When you move positive charge, you create electrical potential differences. You're going to be raising the electrical potential over here in the basolateral extracellular fluid by dumping positive charge there, and you're going to be lowering the electrical potential in the apical extracellular fluid because you are nicking positive charge from there. Positive charge adds positiveness to the electrical potential in an area. If you stick a positively charged ion somewhere, that raises the electrical potential there. So taking away a positive ion from somewhere is going to decrease the electrical potential there. So we're overall going to create an electrical potential difference between the apical extracellular fluid and the basolateral extracellular fluid. Going from apical to basolateral, you're going to go up in electrical potential. Now, that is then going to be the driving force for the movement of chloride anions, because the chloride anions have been left behind here. They're the counterion to sodium that's also dissolved in the water. So you're creating a negative charge over here by leaving the chloride behind, and creating a positive charge over here, to put it more simply, by moving the sodium over here. And in fact, it probably is best just to talk about it that way rather than use the electrical potential difference concept. So now what's going to happen is the chloride anions are going to want to follow the sodium ions. They're going to want to go towards the positively charged ions here. And the chloride anions move paracellularly. Okay, so the sodium ions here are being moved transcellularly, another important key word here. So this is the transcellular movement of the sodium ions. The chloride anions are now going to move paracellularly following the sodium ions. So the tight junctions are reasonably tight. They can stop, massive great proteins that are within the cell membranes moving past them, but they are not so tight that they can stop a single ion moving through them. So they can't stop the chloride anions, for instance, moving through. They are permeable to chloride anions. So the chloride anions are then going to follow by this paracellular route, moving between the cells, so parallel between the cells. That overall now means that we will move sodium chloride anions into the basolateral extracellular fluid from the apical extracellular fluid. That means now that we'll be raising the solute concentration in the basolateral extracellular fluid and reducing the solute concentration in the apical extracellular fluid. So then what will happen is water will move paracellularly. So both chloride anions and water move paracellularly. The chloride is being moved by the electrical potential difference that we're creating across the epithelium here, and the water is being moved by the osmotic gradient that we're creating across the epithelium here. Okay, so that then is the means by which cells in the terminal distal convoluted tubule and the cortical collecting ducts are actually going to reabsorb sodium chloride and water. So resorb fluid, basically, resorb extracellular fluid. We want to increase this process when aldosterone goes up. So that's the overall aim of aldosterone, to increase this process, increase the amount of fluid resorption in the terminal portion of the distal convoluted tubule and the cortical collecting ducts. Okay, so how is aldosterone going to do this? Well, once aldosterone has been created, it can easily gain access into these cells because it is lipid soluble, so it can pass through the cell membrane and end up in the cytoplasm. It's then going to activate a certain receptor inside these cells of the terminal distal convoluted tubule and cortical collecting duct, and this receptor is known as the mineralocorticoid receptor. And the mechanism of this receptor is very similar to the glucocorticoid receptor, which you might have encountered before. So for short, it's abbreviated down to the MR. And by the way, the mineralocorticoid, I suppose I should explain what this means, and I've spelt it wrong already. Mineral, and this should be the O, I've got it the wrong way around. Mineralocorticoid. Aldosterone is what people mean by the mineralocorticoid. So corticoid, it refers to the fact that it has come from the adrenal cortex, and it's going to affect the mineral reabsorption in the kidneys. So mineralocorticoid just refers to aldosterone. So this is the mineralocorticoid receptor, it's the receptor for aldosterone, and for short I'll just abbreviate it down to MR. So the mineralocorticoid receptor is a receptor that will be in the cytoplasm of these distal convoluted tubule cells and cortical collecting duct cells, and usually it's in an inactive state, and when it's in the inactive state it is bound to a bunch of other proteins, of which the most notable one is heat shock protein 90, which again, those of you who are familiar with the glucocorticoid receptor will recognize that's the one that the glucocorticoid receptor for cortisol, another very important steroid hormone, is usually bound to, and which when it's in the inactive state and in the cytoplasm. So the mineralocorticoid receptor is usually found in the cytoplasm bound to heat shock protein 90, usually abbreviated down to HSP90. So HSP90 stands for heat shock protein 90 here. Now, when the aldosterone, the mineralocorticoid, arrives, it's going to bind to the mineralocorticoid receptor, and that binding of aldosterone to the mineralocorticoid receptor causes the cleaving of the mineralocorticoid receptor away from the heat shock protein 90. So I'll draw the aldosterone just now as a little orange blob here. So this is going to represent aldosterone. What's now going to happen is the aldosterone will bind to the mineralocorticoid receptor, like so. So here's the mineralocorticoid receptor with the aldosterone bound, and when the aldosterone binds to the mineralocorticoid receptor, heat shock protein 90 is going to cleave away from the mineralocorticoid receptor. In addition, what's now going to happen is this complex of the mineralocorticoid with the aldosterone, the mineralocorticoid receptor with aldosterone, mineralocorticoid receptor with the mineralocorticoid, if you like, is now going to go into the nucleus. So let me draw this here. So now it moves from the cytoplasm into the nucleus here. And when it gets into the nucleus, it's going to dimerize with other mineralocorticoid receptors that have also bound to aldosterone. So now what's going to happen is homodimerization. So here are the two mineralocorticoid receptors, and here are their aldosterone molecules bound to them. So I'll colour in the aldosterone once again in orange here, and then here is the mineralocorticoid receptor in red. So you get homodimerization. So homo means same, dimerization means joining together of two things. So this is homodimerization once we've got into the nucleus. And now what's going to happen is this homodimer of activated mineralocorticoid receptors is going to act as a transcription factor. Now remember, a transcription factor is something which changes gene expression within a cell, okay? And they are molecules that are capable of binding to DNA, and this transcription factor here will have a specific sequence of nucleic acids which it recognizes. So a specific sequence, they're usually between 8 and 10 nucleic acids long. I don't know what the specific sequence of nucleic acids which this specific transcription factor recognizes and binds to, but there will be a specific sequence, and it's called the mineralocorticoid response element. So MRE stands for the mineralocorticoid, and I can now just show that I can spell mineralocorticoid. So the mineralocorticoid and then response element, and this is the name for that sequence of nucleic acids. There'll be a double strand of DNA, so it will be the sequence of nucleic acids along with the complementary sequence of nucleic acids alongside that, that this specific protein here, this transcription factor, is capable of binding to. So here's the name for it, even though I can't give you the specific sequence of nucleic acids. If you want, you could go away and look up what the specific sequence of nucleic acids that this transcription factor binds to is. But the concept is, I hope, clear. Okay, so this sequence of nucleic acids will then be found in the gene control regions of genes, of a whole bunch of genes that are all going to have their expression influenced by this mineralocorticoid receptor once it's activated. So to draw a picture then for this, so here is some double-stranded DNA. Now let's have one of the genes that's going to have its gene expression influenced by the activated mineralocorticoid receptor. And I want to stress that not all genes will have their expression affected by the mineralocorticoid receptor, but maybe 100 or so will have their expression influenced. So here is a gene that is going to have its expression influenced by this mineralocorticoid receptor in the activated state. The region upstream of a gene is of course known as the gene control region, and this is the region which determines the expression level of the downstream gene, i.e. how often the RNA polymerase enzyme will actually bind to the promoter region, which is part of the gene control region upstream of the gene, and then begin the transcription of the gene to produce the mRNA, and therefore determine how much of the protein is actually going to be produced. Right, so just colouring this on then. So the gene control region is the region upstream of a gene that controls its expression level. There's a very important part of the gene control region which is immediately upstream of the gene, known as the promoter region. So this portion in orange here, this is the promoter region. This is just one part of the gene control region, and this is the region where the general transcription factors and then the RNA polymerase II enzyme actually bind on top of this and assemble. So this is where the RNA polymerase II enzyme binds effectively and then works its way along the downstream gene. So the promoter region is a very important part of the gene control region, but there are then there's more gene control region upstream of the promoter region, and this can then contain some of these response elements for specific transcription factors, as they're called. So I've mentioned a additional piece of information here that now, since I've mentioned it, I'll discuss. So there is a difference between specific transcription factors and general transcription factors. The mineralocorticoid receptor is a specific transcription factor, not a general transcription factor. The general transcription factors, there aren't very many of these. These are things like transcription factor 2D and things like that. They are the ones which bind to the promoter region, which is found upstream of every single gene in the genome, and they have to bind there to allow the RNA polymerase II enzyme to bind on top of them. So whenever you begin transcription of the gene, you have to get the general transcription factors binding, and then RNA polymerase II binds on top of that. Without the general transcription factors, no transcription. There's very few of those. In contrast, the specific transcription factors, there are loads of these, and the mineralocorticoid receptor is an example. These are the ones which will have some sort of response element which they're capable of binding to, in this case the mineralocorticoid response element, and this will be found in the portion of the gene control region upstream of the promoter region in certain genes. So for instance, we might have our response element for the mineralocorticoid receptor located in the portion of the gene control region. So all of this is supposed to be the gene control region for this gene. This is a portion of the gene control region far upstream of the important promoter region, and this might be where the mineralocorticoid receptor dimer can now bind here, and it will then influence the likelihood that the downstream gene is going to be transcribed. And there are different ways it can do this. It can either directly bind to the assembling general transcription factors and stop them from actually correctly assembling, which would then disrupt the transcription of the downstream gene and reduce it, or it could bind and promote the assembly of the general transcription factor complex and then the RNA polymerase II on top, and that would of course increase the expression of the downstream gene. Alternatively, it can change the chromatin structure around this gene. It can make the DNA wrap less tightly around the histones, opening it up, making it more likely that the general transcription factor complex and the RNA polymerase II is going to assemble on top of here. Or it can do the opposite, make the DNA coil more tightly around the histones and therefore close the DNA down, make it less likely that general transcription factor complex and the RNA polymerase II is going to assemble on here. So the basic principle is that the promoter region is this portion of the gene control region immediately upstream of the gene. That's where the general transcription factors all have to bind, and then RNA polymerase II binds on top to even initiate transcription. Okay, so they're very, very important for transcription full stop. The gene control region, however, is much bigger than just the promoter region, and the portion of the gene control region upstream of the promoter region contains response elements, these specific sequences for different specific transcription factors. A specific transcription factor has a specific response element, a specific combination of nucleic acid which it recognizes and combines to a specific sequence of DNA that it combine to, and these response elements, as they're called, will be located in the gene control regions upstream of the promoter region in a bunch of genes, but not all of them, whereas the promoter region is located in all of them, and the general transcription factors bind the same in all of them. And the binding of the specific transcription factor to its response element upstream of this gene can then influence the likelihood that the gene is actually going to be transcribed by affecting the likelihood that the general transcription factor complex and the RNA polymerase II is going to actually assemble on top of the promoter region, either by interacting with it directly or by doing chromatin structure changes. So the overall message here, we've gone a bit into more epigenetics than I was hoping to go into, but I hope that this is clear. The overall message here is that aldosterone arriving in these renal tubule cells of the terminal distal convoluted tubule and the cortical collecting ducts is going to change gene expression within these cells. And specifically, some of the genes that are going to have their expression increased, and just another epigenetic point here, a certain specific transcription factor might increase the expression of some genes and decrease the expression of other genes. It's not the case that they all have to, one has to activate all genes or repress all genes. They can have different effects upstream of different genes. But some of the genes that are going to have their expression increased by the activated mineralocorticoid receptor in these cells are, of course, going to be the sodium-potassium ATPase, okay, and of course the ENaC channel. So you're going to increase the expression of the sodium-potassium ATPase and the ENaC, which are of course the two proteins involved in the sodium chloride reabsorption. They determine the rate at which sodium chloride reabsorption occurs. So if you have more of those, then of course this process is going to occur at a faster rate. You're going to reabsorb more sodium chloride. But of course, essentially, when you increase your levels of these, you're going to have to make sure that they end up in the correct membranes. You're going to have to make sure that the sodium-potassium ATPases all end up in the basolateral membrane and the ENaC proteins all end up in the apical membrane. Okay, so overall then, that is how aldosterone going up is going to increase the amount of sodium chloride and therefore water that is reabsorbed by the terminal distal convoluted tubule and the cortical collecting ducts. Okay, and that then is going to help bring blood volume up. Now, in contrast, if mean arterial pressure is too high, we want to lower blood volume. So that means that aldosterone level is now going to fall below what it would be normally. That means that gene expression of sodium-potassium ATPases and ENaC is going to go below what it would be normally. And note, these proteins have a high turnover, so you are continuously destroying them and replacing them with new ones. So if you reduce the gene expression of these, then you're going to shift the equilibrium, and you're going to be removing these, but then not replacing them at the same rate as you would usually be. So the level of them will gradually go down from what it would normally be in the apical and basolateral membranes. So that means that less of the sodium chloride is going to be resorbed from the apical extracellular fluid into the basolateral extracellular fluid, and therefore less water is going to be reabsorbed. So you're now going to be excreting, excreting more fluid into the urine. So you'll urinate more, and this will result in a reduction in blood volume. So, this is the means by which the renin-angiotensin-aldosterone system then actually produces changes in blood volume in response to changes in mean arterial pressure. Okay, right, so let's now summarize the renin-angiotensin-aldosterone system. So firstly, for mean arterial pressure has gone too low. If mean arterial pressure goes too low, then we activate the renin-angiotensin-aldosterone system. We take its level of activation up above the basal level. That means we create more angiotensin II overall. So I'm not going to go through all the details, I'm just going to put the basics there. And we raise angiotensin II. That's going to have that effect of increasing the contraction level of vascular smooth muscle cells all over the body. So in the venous system, remember, that's going to increase mean venous pressure and help to bring cardiac output up. In the arterial system, that's going to contract arteries, which is going to directly help bring mean arterial pressure up. And in the arterioles, increasing contraction is going to help us to bring total peripheral resistance up, which will help bring mean arterial pressure up. In addition, raised angiotensin II leads to raised aldosterone level, and raised aldosterone will lead to increased water retention by the kidneys and therefore help to bring blood volume back up, which will then increase mean venous pressure, increase cardiac output, increase mean arterial pressure. In contrast, if mean arterial pressure is too high, then the renin-angiotensin-aldosterone system's activity is going to go down. That means that angiotensin II level is going to go down, and therefore the contraction of vascular smooth muscle cells all over the vasculature is going to go down. In the venous system, that will lower mean venous pressure, lowering cardiac output and therefore lowering mean arterial pressure. In the arterial system, that will mean the arteries nicely relax, which will directly help to bring mean arterial pressure down. With regards to the arterioles, they will now all vasodilate, and that will decrease total peripheral resistance and therefore help to decrease mean arterial pressure. In addition, angiotensin II going below what it would normally be will lead to aldosterone level going below what it would normally be, and therefore the kidneys will resorb less fluid than they normally would. It'll lead to diuresis effectively, which means increased urine production. So diuresis means the secretion of fluid by the kidneys, so we're going to get increased diuresis. And that will lead to blood volume going down, and therefore mean venous pressure majorly going down, and therefore cardiac output going down, and therefore mean arterial pressure going down. Okay, so overall then, we have now completed our discussion of the control of blood pressure and blood volume. This is really important physiology to understand, so I hope you have taken a lot from this video.